Welcome to science class. Today we are going to discuss genetic engineering. This is a huge topic with extremely important implications for the future. Technology allows us to wield unbelievable power. That power can be used for good, or it can be used for evil. All technology has that potential. Unlocking the secrets of nuclear physics created the nuclear power plant, which can almost single-handedly rescue us from fossil fuel dependency. And it also created the nuclear bomb, which can almost single-handedly wipe out all complex life on planet Earth. The same technology, but two very different fates. I'm not going to try to predict the future in this video, and I'm not going to say what we should or shouldn't do. My job is to provide you with good information, because we can only make the right decisions if we are well informed. Let's get started. Genetic engineering is the manipulation or change of an organism's genes using technology. Genetic engineering is not a future probability. It's with us today. It's been with us for thousands of years, in fact. Well, it depends on how you view the definition. The ability to directly alter the DNA is a 21st century technology, but we've been able to manipulate gene expression for a very long time. Here's a question. Do you think you have eaten any genetically modified foods this week? The answer is not only yes, but most likely 99% of all the meals you've ever had in your life contained at least one genetically modified food. Another way to think of genetically modified organisms, GMOs, is that they are organisms that expressed genes that they could never have evolved through natural selection. Instead, they were produced by artificial selection through us. Many of the fruits you buy don't have seeds. They couldn't possibly continue to exist in nature, except that humans grow them in a way that allows us to keep getting crop from them. While the majority of the most important crops for humanity rely on natural pollinators like bees, those crops still couldn't exist if it weren't for humans, at least not to the extent to which they do. Many of these crops require intense fertilization and irrigation, which the crops don't get in nature, which is why we have to do it ourselves. It's the same story with livestock. Many of them can't survive without us because they've become too dependent, and in some cases too stupid, to live without us. We grow crops and raise livestock in order to turn them into food. No organism could possibly evolve characteristics that benefit another species without receiving some benefit themselves. Now, it is true that our crops and livestock do get benefits, habitat, free food, protection from predators, and they have evolved a trait that helps them get that, which is tameness. But by and large, we are cheating the system. The GMO acronym is used as a boogeyman by some people. GMO equals bad. The fact is, if we eliminated GMOs from the planet, billions of people would starve to death. We could not possibly grow enough food to feed the current world population if we did not use GMOs. Selective breeding and other technologies have allowed us to turn corn from this into this, berries and fruits from this to this, chickens from this to this. Most people think if it wasn't created in the lab, then it's not a GMO, which I don't agree with, but we will focus on more recent technologies from now on. There are a few major lab techniques for altering genomes. We will focus on recombinant DNA techniques and CRISPR. Recombinant DNA is a technique where you introduce new DNA to an organism. It's similar to horizontal gene transfer, but it's done by humans. There are a few ways to make recombinant DNA. One is to introduce the new DNA to a cell and then have that organism express that gene. We can harvest bacterial plasmids and then force a transformation in the bacteria to occur. Another method is to insert the desired gene into a virus and then purposely infect the cell with the new DNA. It's also possible to make copies of the gene in a test tube using the process of polymerase chain reaction, or PCR. But the genes made in the test tube still need to get introduced to the organism. Now you may be asking yourself, why would you want to do this? Well, I'll just share with you some of the things we've been able to do using recombinant DNA techniques. Some bacteria have a gene that produces a toxin called Bt toxin. This toxin kills insects, but it's harmless to plants, humans, and most other animals. Crops have been given this gene, and when bugs eat the plant, they die, so there's less need for chemical pesticides. Around 30% of the milk you buy comes from cows that have been given hormones that were made by bacteria in large quantities to boost their milk production. Pigs have been genetically modified to produce more meat than fat and to produce more omega-3 fatty acids, which are more healthy than omega-6 fatty acids. More than 7 million Americans need to take insulin, which is normally made by the pancreas. Now you can donate blood, but have you ever heard of somebody donating insulin? You haven't because you can't, or at least you don't need to. Decades ago, we had to extract insulin from cows and pigs. It was extremely laborious and expensive, and you did not get much insulin. Today we have injected bacteria with the gene that codes for making the insulin protein, and now we make huge quantities of insulin inside vats. It's almost like the way we make beer, except they're making insulin. People with hemophilia can get the proteins they don't naturally have thanks to the same technology. This has saved a huge number of lives. Some vaccines are also made using recombinant DNA technology. PCR technology is what led to the DNA revolution in forensics. Now that we can make copies of DNA, we can run low-tech but highly accurate tests on DNA. 110 people in the United States have been rescued from death row since the 1980s, thanks to this technology. PCR is also used in paternity testing, and it has led to a huge amount of entertaining but cringe-worthy moments from television.
Andrew, you are not the father.
Applying gene modifications directly to humans is not yet a wide-scale practice. Jesse Gelsinger underwent an experimental gene therapy treatment in 1999, but he died shortly afterwards of anaphylaxis, where the entire body has a simultaneous allergic reaction to the virus he was given to introduce the new gene. Since then, we have hit pause on these types of gene therapy techniques, but that won't last forever. I feel that we should pursue these things, if nothing else but for the sake of pursuing knowledge. Because, as I said, we can't hope to make the right decisions unless we are well informed. The future of genetic engineering appears to be CRISPR-Cas9. To make things simple, what CRISPR allows you to do is pinpoint gene editing. For example, CRISPR can find a faulty gene, cut it out, and then replace it with a healthy version of that gene. Technologies like CRISPR could allow us to one day eliminate almost all genetic defects, curing diseases that have a 100% fatality rate, such as Huntington's. It could potentially allow us to add to our genomes as well. The possibilities are almost endless, as well as the potential to do both good and evil. Well, that concludes our videos on genetics and DNA. Next time, we are going to begin studying evolution. I am extremely excited. Thanks for watching.